Hey, good afternoon. We'll get cracking. We had quite a few registrations, and I appreciate the circumstances today are quite hectic and very different again. But nonetheless, thank you guys for taking the time to join us for this webinar. So I hope you're all well to begin with. I appreciate that we're due to start in the UK what has been known or branded as Lockdown 2.0. I know that means that there's more uncertain times ahead. So I hope you guys all remain well and keep busy over the next couple of months. And we'll soon be Christmas before you know it. So for those of you that have seen the PDF advertising this webinar, which had the link on, you may or may not be aware that if you sit for 80% or more of this webinar, you will receive CPD points, and you get a certificate of that from Tecniplast following on from this webinar. It will be recorded. So as always, we ask please respect the speakers and keep your mics on mute. You should have the option to open a chat box. You can chat openly to everybody if you wish to raise a question, or if you prefer, ask it privately to myself and I can manage that question without naming names. But please feel free to ask questions throughout, and then we will address those at the end of the two presentations. If you could just put the name of who you wish to direct the question at, that would be very helpful. And that's that. If you have any issues with that, again, just mention to me on the chat and I'll talk you through how to do that on this platform. Another quick sort of tool, if it makes it any easier, particularly when the guys have got some data on the screen, when you hover your cursor over the presentation, you should be able to zoom in and out, allowing you to see things a little clearer if it helps. So in terms of structure then, after my introduction, I'll turn the camera off, but I'll introduce you to the guys with an image, just so you know who's talking and who's presenting their data if you've not already met them. And then I'll hand over to Joe to present. Then we will immediately move on to Judit, and then we'll take the questions afterwards. So thanks for your patience if you've got any questions to wait until the end. So as the webinar details highlighted, this is looking at monitoring mouse activity to highlight the impacts of disease progression and improve animal welfare. These tools that are very much growing and more of a talking point are now able to access and open more information 24/7, both on the scientific side and the research, as well as providing more welfare information. So here are the guys that are going to be talking today. So we've got Joanna Moore from GSK, who been quite intensely using the DVC in the past few months, and she'll be focusing on home cage technology, which she's continued to build on her research for the welfare of animals in order to help researchers to further understand the motivation and needs of animals in their care. She's been working with laboratory animals for over 20 years, with a special interest in their welfare and care. I know she's heavily involved in that at GSK, not just in the UK, but across other sites. And Jo has been working in academia and industry and was awarded a PhD in animal sciences in 2018. Following on from that, we have Judit, who will be presenting. She's a senior researcher from AstraZeneca. So she will be focusing on aspects around the actual disease progression and the scientific data behind that. So Judit was born in Barcelona, where she obtained a degree in biology and a PhD in neuroscience at the Universitat Autònoma de Barcelona. After completing a PhD, Judit moved to Cambridge to join the CRUK team, where she worked on developing in vitro and in vivo models to study renal cancer. From 2017, Judit has been part of the Animal Science and Technologies team at AstraZeneca. Her work is focused in assessing the efficacy of newly developed drugs and evaluating and refining new and old in vivo models for oncology. And then finally, we've got Stefano Gaburro with us, who's a scientific director at Tecniplast. So he's here to facilitate and answer any particular questions around the platform that's been used. He's worked closely with these guys on data management and also been involved in writing his own papers in the past. Just to briefly mention at the beginning that only this week, Stefano's had a review paper published, which the link is here. I'll copy and paste this into the chat box. More than happy to share it at a later date. And this is called 3 Pillars of Automated Cage Phenotyping of Mice, Novel Findings, Refinement and Reproducibility based on Literature and Experience. So this is quite a comprehensive review. Sounds a bit contradictory, but it's quite an interesting way of looking and comparing multiple home cage monitoring systems, being that's the way the world seems to be moving, and looking at the benefits that those might bring to various fields of science. Just thought it's quite timely to mention that as this was released this week. Well done, Stefano. So now I'll end my welcome and I'll introduce Joe. So hopefully you could share your screen, Joe, and we can start the first presentation.
Okay, thanks, Scott. Thanks for inviting me to give this presentation. And today I'm going to talk about the second of 3 studies we carried out with this programme of work looking at timed mating. Today I'm going to be talking about a study we completed using home cage monitoring during timed mating procedures to determine the impact of swapping a female versus adding a female to an established pair's cage. For this study, we used a strain of mice that produce naturally sterile hemizygous males called prim1s. If you're interested in what a prim1 mouse looks like, then this is an example of one. And to identify a prim1 mouse, we don't need to genotype, as we can tell by the coat colour. The males and females are always agouti, and the extremities are bright pink because they express a red fluorescent protein. Although only the males are sterile. And if you're interested in learning more about this strain, please look at the paper in my reference list at the end of the slide set. All animal studies were ethically reviewed and carried out in accordance with Animal Scientific Procedures Act 1986 and the GSK policy on the care, welfare and treatment of animals. The use of a naturally sterile male is already a good refinement for us, as we no longer have to use surgery to create them. We further refined this model by also always housing them with a companion female, as the males are often kept for up to 9 months. And these males are used to induce pseudo-pregnancy in the female mouse, and the females tend to be swapped in the morning in line with the daily routine. They are then left with the male overnight, usually for a 24-hour period. The use of the term action units is a commonly used term now and reference to the rodent facial expressions in the Grimace school, which is generally used as an indication of pain in rodents and other animals. And in a similar way, we can break down the disruptions in activity to the animals into disruption units. Some disruption units are unavoidable, such as when we go into the room and when we do the animal welfare checks in the mornings. However, there are several we can remove from timed mating procedures, for example, cage changing and swapping the females. So for this study, we removed as many of the disruption units as we could from one of the groups of mice, leaving them with only 2 sources of disruptions or interventions. When we placed the new female in the cage with the established pair, and when we removed the new female at the end of the study. When we cannot be certain by visual assessment alone what the impact a cage intervention has on cage activity, there are publications which have shown that high increases in cage activity are related to disruptions caused by other potentially high activity generating interventions, such as cage changing and procedures. For example, Pernoa et al. Suggested that procedures involving handling or the transfer of the animals disrupts their basic activity pattern and induces changes that appear to impact them over an extended period of time. We hoped that using home cage activity monitoring might help give us an objective answer to 2 different timed mating scenarios. Firstly, swapping a companion female for a new female, and secondly, adding a new female to an established pair's home cage. We hypothesised that the addition of a new female to an established pair would cause less disturbance for the male mouse. Therefore, activity would return to a typical pre-change levels in a shorter time, compared to the activity for males that have had their female substituted for a new female. But the only way we can visually compare cage activity between uneven groups is to normalise data by the number of animals per cage. And when we normalise data by animal numbers to compare different group sizes, it's really not ideal, and any results generated in this way should always be viewed with caution, which includes the results I'm going to talk about today. Our overall aim was to show whether there was an observable change in cage activity when a companion female is replaced with a new female, compared to when a new female is added to a cage. Our main objective was to visualise and compare the cage activity in two different new female mice timed mating scenarios. First, I wanted just to talk a little about working with big data, because as Scott said, this was one of our very early studies using the DVC. And with so much data generated, it's extremely important to construct your scientific question prior to the start of the study. Otherwise, you may find yourself overwhelmed with the amount of spiky data you can see, and the genuine results can be hidden amongst all of the noise. We learned a lot about how to work with large amounts of data during these early investigatory studies, and realised we had to adapt the experiment to the DVC so that we were using it properly and would not get a false negative or a false positive result, or even worse, a very unexpected answer to our research question, which may lead to incorrect results. And one of the benefits of the DVC is the data from it can be downloaded via the analytical platform to a readable Excel file. It's relatively easy to select the timeframes of data you want to download, or the days or dates or months of it. And one of the other benefits of the analytical platform is that it keeps data, all of the data generated from every cage on the rack, so that we can carry out further investigations of data long after we have completed one study. The DVC itself is a non-intrusive tracking system, and it sends data. The DVC we use sent data from the rack to a cloud storage via the master computer. It's important to note, though, that when there is more than one animal in the cage, we always consider the cage as the experimental unit. And the data is the animal activity index measured across all 12 electrodes per minute. 0% means the animal is not moving at all over the observation period, in this instance 1 minute, and 100% means the animal was able to activate all of the electrodes for the entire minute of the observation. The 100% value is impossible to get, as it means all of the electrodes are activated for the entire minute. And as you can see in the picture on the right, I've drawn where the mouse might be activating the electrodes, which is the orange is the outline of where the electrodes are on the cage. And it's just impossible for him to activate all of the electrodes in 1 minute's observation period. The 100% value is therefore impossible to get, as all of the electrodes would be activated, and the animal movement is activating all of the electrodes and changes the status to active rather than inactive, and vice versa for each electrode it crosses. The system is calculating the overall activity on the board and averaging the time period you're using. If you aggregate by an hour, it's 60 times 60 times 4 samples, or as we did in this study, if you aggregate by minute, it is 60 times 4 samples. Therefore, the summary average activity is the average over 1 minute, which is the aggregation of time we used. It is important to have a reasonable understanding of the baseline activity of the mice being studied to determine what activity period will give the most accurate answer to a scientific question. After initially reviewing these mice, we found that they had a very sporadic activity across the day and night periods compared to other strains. This might be related to the social interaction within the pairs and trios, especially as they were mixed sex groups. And to compare the uneven cage occupant numbers by animal number, as I said, prior to carrying out our analysis. The use of normalization by animal number does have limitations of data interpretation, but it can enable broad inferences of the data, although it can't give a precise answer to a scientific question, which is why we have to treat it with caution. This study ran over 5 days, Monday to Friday, including 20 cages of established PRIM1 pairs and 5 cages of established PRIM1 trios, with 5 cages of 4 CD1 females, which were weight matched to the companion females. All our mice were housed during the study in a GM 500 individually ventilated cages on the DVC rack. Each of the cages contained our standard enrichment for all of our animals, and it's worth noting that the mice used in this study were housed under a light cycle of 14 hours light and 10 hours dark, with a 10-minute dusk and dawn period. The reason for this is they were housed in our mouse breeding facility, and of course they had food and water available at all times. So we included 20 established pairs of PRIM1 mice and 5 established trios of PRIM1 mice for the controls trios. We had a further CD1 weight matched females, and these were included for the new female cohort. The cages were randomly allocated between 2 groups. 10 pairs were in group 1, where the companion female would be swapped for a new female, and the remaining 10 were put into group 2, where a new female would be added to the established pair. The control cages were established trios, which acted as controls for group 2 only. We already had some control data for pairs from our previous study. The data ran over a 5-day period, as I said, Monday to Friday. On day 1, all of the study animals, including the controls, were transferred to clean cages. On day 3, each male mouse in group 1 had 1 companion female replaced with a weight-matched new female. And for group 2, each of the cages of mice had a new female added to the established pair's cage. The control females were removed from the rack, and each female removed briefly for up to 5 seconds and put back in the home cage. And on day 5, the study was completed, and all of the new females that were used were replaced with the original companion female, or removed from the established pair. Each of these procedures took roughly about 10 seconds per cage. We focused on the activity at the following 3 2-hour time intervals: 10:10 to 12:10 p.m., 12:10 to 14:10, 1400 hours, and 8 o'clock till 10 o'clock at night. We took an average of the preceding and subsequent 3.5 hours for each 2-hour time interval and looked for trends in the data. We then looked at the activity in the 2-hour time interval directly after female replacement or addition. For each 2-hour time interval, we calculated the change from baseline for each cage to determine the activity increase or decrease on the day of the female replacement, compared to the previous days and the subsequent days for the same time interval. And these were our results. Initially, I wanted just to show you the heat map. The control trio's heat map is at the top of this slide, and group 2's, where the female was added, is the middle heat map, and group 1, where the female was replaced, is the lower heat map. It's important to note the aggregation of this heat map is every 5 minutes rather than 1 minute aggregation, which we used for the study data. This was just to make it easy for me to visualize on a PowerPoint slide and to show you the activity patterns clearer. If you look at box 1 showing the activity after cage changing, you'll see that we did not see the intense reaction to cage change disruption that we have observed, as I'd mentioned, with other mice and which has been mentioned in other publications. In the heat map, we can see that the blue, which indicates the animals are not moving and likely resting or sleeping, there is not much blue between the lights coming on and the cage changing. This suggests that mice were active prior to the cage changing commencing. And if you look at the activity in box 2, where the female was replaced, we can see the extended activity period caused by the addition of the female in group 2 and the female replacement in group 1, which we have observed in other similar studies. We can also see that the tail end activity is longer for group 1 compared to group 2. These plots show the average activity over time frames for group 2 trios versus their control trios. And the plots are the results of this exploratory study where we compared group 2 when the female was added with the trio controls. Although this is just for the trios. But we're looking at the highest activity peak of 10:10 to 10:12. The first graph shows the pre-intervention activity between group 2 and the control trios was similar. However, when a female was added, there was higher activity in group 2 compared to the controls on the day of the intervention. On the subsequent day, we can see the activity becomes much closer to the control trios. The cage in position, the outlier, D5, is an indication that the cage was in position column D and row 5 on the rack. These plots are the post-interventions, group 2 trios versus the control trios. And these plots show the average activity of group 2, where a female was added to an established pair, compared to the control trios across all time frames tested, between the day of intervention and the subsequent day. These graphs show a gradual reduction of activity over the time intervals rather. Activity was higher between Wednesday and Thursday for group 2 during the 10:10 to 12:10 p.m. 2-hour time interval. Aside from one outlier, there was no difference really between group 2 and the trio controls between Wednesday and Thursday during the 12:10 to 14:10 2-hour time interval, with both groups showing low activity. And activity was roughly the same between Wednesday and Thursday for both groups during the 8 p.m. to 10 p.m. 2-hour time interval, which is when we expect the mice to be active. These plots show the overall results of this exploratory study, comparing the group 1 prior to the female being replaced with group 2 prior to the female being added, with the outlier cages once again indicated. The graph shows that on day 1 and 2, activity was at an expected level for these mice across all of the timeframes we examined, and a direct comparison wasn't possible after Tuesday due to the uneven groups without normalising the data. But here we can see the animals are all looking as we expect. The post-intervention, group 1 compared versus group 2, the plotted data was initially normalised by mouse numbers per cage. In these plots, we carried out a direct comparison between group 1, where the female was replaced, and group 2, where the female was added. These plots show the average activity between group 1, where the female was swapped, and group 2, where a female was added to an established pair. During the 2-hour time interval of 10:10 to 12:10 p.m. on the day of the intervention, compared to the subsequent day, group 1 shows a small increase in activity compared to group 2. In the 2-hour time interval of 12:10 to 14:10, group 1 appear more active compared to group 2. And in the 2-hour time interval of 8 till 10 p.m., the activity for group 2 increases from Wednesday onwards, whereas the activity for group 1 decreases. We can see this more clearly in the activity of group 1 compared to group 2 in this slide set. During the 10:10 to 12:10 p.m. 2-hour time interval and the 12:10 to 14:10 2-hour time interval, and we can see the visible decrease in activity in 8 o'clock till 10 p.m. time interval in group 1 compared to group 2. This slide shows the same data as the previous slide, but in a much clearer way. So what can we additionally infer from this study? Well, we could see that cage changing did not appear to cause the large spike in activity we've seen in our previous studies and in other published studies that have used home cage monitoring to look at how disruptions can interfere with animals. However, in the heat map I showed you earlier, we could see that the mice were fairly active prior to the cage change at 8 o'clock, which meant that there was human activity in the room earlier in the day. And when I looked back, sorry, the cage change was at 9. When I looked back, over the graphs and the data, I could see that the room had been entered at 8 o'clock, and that was when we had started removing cages to check the animals. This meant that there was human activity in the room in the lead up to the cages being changed, likely meant the mice were quite awake and not resting at the time of the timed mating. The ability to align what has happened in the room to create potential spikes in activity can help us understand the reasons behind unusual peaks in the activity or study results that we observe. We can also use the heat map to review activity patterns of animals prior to studies to determine whether we can carry out interventions when they're most likely to be active, especially in animals like the ones I've talked about today, because they had such a spiky activity pattern. They're often naturally active at several time points throughout the day. If we can carry out routines when they're naturally active, we could further reduce the impact on their circadian rhythm. During the initial study, the time mating procedure started almost as soon as we entered the room, and the increase in activity caused by the female was still visible even though we carried it out at the same time as cage changing. There was a huge spike in the activity of the group that had the female replaced at the same time as cage changing compared to the group that only had cage changing. But there was big increases in activity in cage changing, much more intense activity on the heat map compared to the heat map we're seeing on this study. So maybe by allowing the animals time to adapt to people being present and removing some of those disruption units, just such as cage changing, it could be possible to reduce the impact of timed mating on mouse activity spikes. And it may be that reducing the disruption units will further impact the procedures on activity and general cage disruption. To compare the uneven cage occupant numbers, we normalise the data by animal number prior to carrying out our analysis. The use of normalisation by animal number has limitations of data interpretation, as I said. And while it can allow the broad inferences from the study, which we've made today, it cannot give a precise answer to the scientific question. Therefore, results must always be interpreted with caution, and we're planning further studies to confirm the results we've found, because we're pretty confident there's something going on here. We looked independently at the response of cage activity when a new female was added to a compatible pair, consisting of a sterile male and companion female, and found there was no clear reduction in mouse activity during the 8 to 10 p.m. 2-hour time interval. And if you remember, the 8 to 10 p.m. is when our lights go out. 8 o'clock is when our lights go out, because it's the breeding unit. This may be due to the lower disruption we observed in this group during the 10:10 to 12:10 p.m. 2-hour time interval, compared to when a female was replaced. This study indicated that there was a lower activity response in the 10:10 to 12:10 p.m. 2-hour time interval with a new female, when a new female was introduced to a compatible pair compared to when we exchanged the females. This increase in activity for the replacement female group suggests that there may have been a higher disruption on the normal daily activity of these mice as a result of the female replacement. The spikes in activity after the female swap may be related to the male searching for his former companion and the new female searching for a mouse that is not there. Whereas when a new female is added to an established pair, there may be less non-mating activity in the cage, possibly. If you've seen my previous webinar of timed mating, you will have heard me say that in our subsequent study, we found that there was a reduction in mouse activity during the 8 to 10 p.m. 2-hour time interval, the day of the female swap, compared to their non-swap control pairs. Again, we put this down to the mice being possibly fatigued during the dark phase, due to the huge spike in activity at the 10 a.m. to 12 p.m. 2-hour time interval. And taking both these results into account, there is an indication that the disruption caused by swapping females is higher compared to adding a female to an established pair. Looking at this study, though, on its own, it's hard to know with any certainty whether levels of activity return to pre-swap or addition levels of activity when we're comparing different numbers of animals. But it does give us an indication we could do more to reduce the high activity created by time mating, which could in turn remove the fatigue we have observed in the mice during the dark phase. As it is generally accepted, activities which affect the circadian rhythm can have an impact on welfare, and it may be something as simple as replacing the female at the end of the day or adding the female at the end of the day will lead to cage activity returning to pre-swap or addition levels sooner. As always with any of these studies, there's always a lot of further work and questions to be asked. And for this study, our assumption was that while the behaviour exhibited at the time in the time mating scenario may be beneficial for the male, putting him through this procedure on a weekly or twice weekly basis may not, due to the potential impact on his overall activity across a 24-hour period or the weeks. And this meant the male has been the focus of these studies. The cage, though, is the experimental unit, and therefore all the animals within it have to be treated as one. How we could overcome this is to draw out individual mouse activity. To do this, we could use a DVC metric for pair housing, which calculates the distance when both of the animals are moving, which is a good biomarker for the social study. And while this can inform us of the difference in an individual pair-housed mouse activity, it still will not inform us on which mouse is creating the activity and what behaviour is causing the activity. If we added a video, we could see what activity spikes were being caused by and potentially which of the mice were causing it, especially with these animals, as the prim1 males are agouti and the new females are all albino because we use CD1s. So we could therefore use the coat colour to identify them. But if we use the video alone, we'd have to view the full video to see where the spikes of activity were. And another addition to this could be using an ultrasound monitor to determine whether the squeaks, which I've been told are mainly produced by female mice, are a positive sound or not. But in a similar way to the video, we'd have to listen or look at the whole recording to find the peaks. However, when we use the DVC, we would only need to check the video and ultrasound recording when there are peaks on the DVC, because these can be linked, because the DVC can be linked to other electronic devices. Another platform connected to the DVC is called the Workplace platform. Here we can see when each cage is removed and replaced on a rack, which technician has carried out a procedure and any alarms and why the alarm was generated. This means we can look at data on this platform to see what might have caused any unusual activity. And if we are able to use all of these technologies, it would give us a more holistic overview of what's happening in the cage. Although, the more we find out about animal activity and the complexities of behaviour that we discover, the more we realise how much more we have to learn about these animals and how much more we need to do to improve their well-being. Due to the number of cages we had on the DVC, we had to use a standard IVC rack to house the females when they were not used on this study. So we were unable to go back and review their data. Yet home cage monitoring produces activity data we really cannot obtain through observations alone. So we really missed out on a lot of information in this study, because our initial study was designed only around the cage the male was housed in. We didn't see any of the female responses to their own different scenarios. It would be really interesting to follow the companion females and see how they react to being put in a new cage with other companion females. Are they less active than they would be if they were put with a male? Do they fight? Is there an intense activity when they first get put in, which is then shown as a more fatigue in the dark phase? We just don't know the answer to these questions. And equally, what happens when they return to their original companion male? Is there still smells and cues that would enable them to remember each other, and therefore the impact on the cage activity is lower when she's returned? Or do they behave as if they've not met before, and therefore the activity is higher, with again the reduced activity in the dark phase? And even when you do an activity you enjoy that takes you out of your circadian rhythm, we all know it can take us quite a while to get back into the rhythm and feel much better about things. If you're very tired, it can make you quite grumpy. Another area we have not observed is the impact on the new female, both when she's removed from her cage and her cage mate and placed with the male, and when she's put back with her original group. We don't know what these activities are. And if we could look at the activities on the female as part of a holistic overview, it could give us even more insight in the impact of time mating on our mice and give us more information on how we may be able to refine it. After we were able to see changes in the activity of the animals and a possible reduction of the disruption as a response to cage changing in this study, which we did not observe previously, it would be good to test whether setting up timed mating at the end of a working day will not only reduce the disruption at the time of the mating, but also remove some of the changes we observed in cage activity during the dark phase. Several studies have shown that carrying out studies and procedures under reverse lighting would have a benefit for the animals, and this is another area we'd like to investigate in the future. And it could include things like cage changing and other activities in the room. So overall, home cage activity monitoring has given us a unique insight in terms of how the work we do can impact the welfare of animals, thus giving us an opportunity to refine our husbandry procedures and processes, the scientific studies and care to further meet their needs. And it's worth bearing in mind that keeping animal numbers consistent between each cage will help quantify the trends better and give us a much more precise answer to any scientific question. I just wanted to thank all of the people involved with this project, especially Guido, Fabio and Scott from Tecniplast, who gave us some really great support and help in learning and understanding how to work with the DVC and the data. A very special thank you to Eloisa and Julia at GSK, who gave me amazing help and statistical support. And finally, I'd like to thank you all for listening, and do ask any questions when Scott tells you to. Thank you.
Thanks very much, Jo. As always, you get a series of quite robust data that then leads to more and more questions, but I think you summarise those quite well. So in terms of Jo looking at the system and DVC home monitoring around more welfare, social behaviour and obviously mating aspects, Judith's going to take a slightly different stance in terms of looking at how this technology could be used to cover disease, health and welfare aspects within home cage monitoring. So if you're all set, Judit, over to you, please.
There you are. I think now you can hear me. Okay, so thanks, Scott, for the presentation, and thanks for inviting me to present in this webinar. And for today's talk, I'll be focusing on how monitoring the homemade activity can be used to improve the evaluation of in vivo models and also in vivo procedures. So just sorry, I don't think that's working. There you are. So just a brief summary of this talk. So I'll go through what we consider are the advantages of continuous monitoring of the animal activity within the home cage, and how we have applied this technology as a tool to evaluate the oncology in vivo models, focusing on both flank tumor model and orthotopic pancreatic model. And finally, I'll present some work done in how to apply this technology to assess animal welfare after invasive procedures. Now, what are the advantages of having this 24/7 monitoring of animal activity in the home cage? So I won't get into the details on the technology behind the DVC rack. I think Joe has already nicely commented to that. I just wanted to mention how the housing in the DVC rack compares to the green line conventional caging that is widely used in the animal facilities. And as you can see, the differences are minimal. The type of racks and caging are exactly the same. So by using this technology, we are not interfering in the normal housing conditions of our animals. What we get with the DVC rack is a constant monitoring of animal activity in an environment that is familiar for the animal, and including holidays, weekends, and what we think is more important, the night period. Now, why do we think that this 24/7 monitoring is important? So, as I mentioned, the DVC rack allows us to monitor the activity in an environment that is familiar for the animal. As you may know, traditional evaluation of animal behavior relies on tests that put the animals in what we can consider as artificial situations, like swimming and other type of tests. Now, since they are based on using this artificial environment, by using only this type of tests, we might be missing some important information from our models. So monitor of normal activity levels with an environment that is familiar with the animal can provide new information on animal behavior and animal welfare. And that's something that's otherwise really difficult to have access to. The second advantages provided by this automated monitoring is being able to avoid human interaction. So for the animals, we are a source of stress. So by avoiding having humans around, we are allowing the mice to express better their normal behavior. And finally, this technology gives us access to the night period. So as you know, mice are nocturnal animals, and traditionally behavioral tests and welfare assessments are done during the day, when the humans are awake, but the mice are usually sleeping. And this is clearly shown by this graph at the bottom that represents the average mice activity of every hour throughout the day of a period of roughly a month. And this is the data obtained by the DVC rack in our facilities. And as expected, we can clearly see that the animals are much more active during the night period than during the day. So if we are aiming to evaluate changes in activity, it makes sense to focus in that period in which the animals are more active and without the human presence or interference. And that is clearly the night period. Now, how having access to the night activity can impact or improve our knowledge of in vivo models. So at AstraZeneca, we focus on developing drugs that will help the patients. And obviously, those patients have diseases. So we mainly work with animals that are modeled in order to mimic human diseases. We have worked under the hypothesis that the disease presence and evolution will alter the normal behavior of the animals since they're obviously feeling unwell. And that can translate in an alteration of their activity in the home cage. This alteration of movement can be predictive of both the progression of the disease and also the response to the treatment that we are evaluating. Besides that, it also provides a better assessment of animal welfare since we are evaluating an objective measure such as the animal activity. So to summarize, understanding the impact of the animal disease in activity can improve the evaluation of the in vivo models and provide better scientific endpoints to our studies, especially in those models in which evaluating the disease progression or regression is not straightforward. So the first questions after acquiring our DVC rack was to evaluate if the progression of the diseases we are modeling have an impact at all in the normal movement of our animals. Initially, we evaluated this by using a simple oncology model that is heavily used to evaluate the efficacy of the new drugs and that allows for a nice monitoring of the progression of the disease, and this is the flank tumor model. This slide shows the design of our experiment. So for flank tumor models, we implant tumor cells subcutaneously in the flank of the animal. And since the tumor grows at the site of the animal, we can evaluate the progression of the disease simply by measuring the size of the tumor using calipers. For this particular study, we used biopsy females that were grouped at 4 animals per cage and were housed in the DVC rack from the moment they arrived to AstraZeneca. After implanting the tumor cells at day 0, we monitor the tumor growth conventionally by using calipers, and thanks to the DVC rack, we had continuous monitoring of the animal activity per cage. At the same time, we had a group of naive animals that were not implanted, and we used them as controls for normal activity. Now, this slide shows the average movement of both groups during the different hours of the day, and that's an average of the movement throughout the whole study, and also separated by day and night period. We have calculated, as I mentioned, the average movement per hour in both the control animals and the tumor-bearing animals, and as you can see, during the day period, that's the middle section of the graph, both groups behave in a very similar way. However, when we focus on the night activity, we see that the tumor-bearing animals, which is the line in blue, show the decreased activity levels when compared to the non-bearing animals that are shown in orange. We did some statistical analysis of this data, and that difference is significant. So this type of information suggests that the presence of the tumor affects somehow the activity levels of those animals. To understand whether we could use the animal activity levels to predict the disease progression, we evaluated if there was a correlation between the disease status and the activity levels focusing into the night period. That's when we saw mainly the difference in animal activity. To understand that, we try to correlate the volume of the tumor of the animals with the activity levels of that same animal at a given time. As I mentioned before, those animals were group housed. Initially, we had 4 females per cage. However, due to the human endpoints that we have in place associated to the model, the mice were euthanized at different time points after cell implant, mainly for welfare reasons. That means that the total animal number per cage varied throughout the length of the study. To account for those differences in housing density, we took the raw activity data provided by the DVC rack as a cage aggregate, and we normalized it by the number of animals in the cage at each given time. And we used that normalized value to correlate it with the tumor size of the animal at that same given time. And as you can see, the graph is showing that correlation, and we can see an inverse correlation between the tumor size and the average movement during the night period. In the graph, I'm showing each of the colors represents a cage. And as you can see, as the tumor increases in size, the activity levels of the animals during the night period decreases. And again, our statistics teams did a great job analyzing this data, and we saw that this correlation is significant. So we were quite happy with that correlation seen. So we moved forward to evaluate the same concept in a more challenging oncology model, such as the pancreatic orthotopic model. In this case, the tumor cells are injected in the tail of the pancreas under a surgical approach. That gives us a more relevant tumor environment in terms of modeling the disease, since the primary location of the tumor is the same as it's seen in humans. It also allows for a better modeling of metastatic disease. However, since now the tumor is growing inside the animal, monitoring the progression of the disease is more challenging, as we rely on palpations to guess the size of the tumors and the presence of other clinical signs that appear due to the evolution of the disease. Another advantage of using this model is that it has allowed us to evaluate the impact of the surgery itself in the animal. So again, this is the design of this study. In this occasion, we use individually housed males, again, housed in the DVC rack from arrival. The monitoring of the disease in this case, as I mentioned before, is by palpation. We palpate the abdomen of the animals 3 times a week, and we evaluate other clinical signs such as the presence of ascites or weight loss to have an idea on how the disease is progressing. This is the graph that is showing the evolution of the disease using our conventional method of scoring based on tumor size and clinical signs. Each of the lines represents a mouse in this study. And as you can see, all the mice follow a lovely evolution of the disease as expected, or as planned at least. So to evaluate any correlation with movement or activity levels of those animals, we took a similar approach as with the flank tumor model. And we try to evaluate the correlation of this disease scoring with the animal activity, again, focusing in the night period. And as you can see, we saw similar results as with the previous model, an inverse correlation of the status of the disease and the average night activity of the animals. So in that case, one of each line represents one animal. And again, the more progressed the disease is, the bigger the scoring of the disease is, we see less and less movement during the night period. Now, understanding this correlation can be really useful in those models in which modeling the disease progression can be challenging and can also be evaluated to be used to evaluate the effect of treatments and to improve human and scientific endpoints. However, when we were doing those experiments, we also learned that animal activity can be influenced by several external stimuli that we initially didn't account for, and those need to be taken into account when using the automated monitoring of animal activity or animal activity in general to evaluate disease progression or disease regression. The most important external influence on activity levels we have seen is the change of the bedding. We knew beforehand that after changing the bedding of the animals, they immediately show an increase of activity levels. You can see it as you are in the animal unit. And that's probably due to an increase of the exploratory activity. Since changing the bedding implies a new environment, they have new toys, so mice will naturally go and explore the new stuff, and therefore we see an increase in the total activity levels. Now, this expectation was confirmed when evaluating their activity 24/7. These two graphs show both the night and day activity of the animals during several. Those are actually the previous animals that I showed the correlation with the tumor size and animal movement. Day 0 is the day of surgery, and as you can see, there's a massive peak both in the day period and in the night period, and that corresponds to the change of the bedding. Now, the problem is that, as you can see, especially in those animals, the peak of activity is quite significant, and this sudden increase in activity can interfere in the monitoring of the disease evolution when we are using the activity levels as a scoring of a disease progression. So I'm showing here an example, again, data that we have obtained in the facility, same animals that I've presented before. And in that case, the bedding change in those animals was changed at the initial phases of the disease. So the day 0 is the surgery day. And as you can see, roughly 10 days after the surgery, they had their bedding change. If we map this bedding change on the progression of the disease, we can see that the bedding was changed at the very initial stages of the disease. So that spike of movement caused by the change of bedding didn't interfere with the potential decrease of activity due to the evolution of the disease. And again, that's another way to map this bedding change. And as you can see, where the bedding was changed, we were kind of expecting high activity levels of those animals anyway. However, almost in parallel to that study, we had another group of mice that also underwent through orthotopic pancreatic surgery to implant the tumor cells. And for those animals, the bedding was changed later on after the surgery. So as in here, roughly 20 days after surgery. Again, if we map this bedding change in the progression of the disease or the disease status, we can see that the bedding in these animals will change when the disease was quite advanced. So at the time the bedding was changed, we were hoping to see a reduction in activity levels induced by the presence of the tumor. However, by increasing artificially, if you want to call it, the activity levels by changing the bedding, that masked the impact of the actual disease in the animal movement. And as you can see in the bottom right graph, we were not able to see any correlation between animal movement and tumor scoring in those animals, because by the time we were expecting the disease to have a detrimental effect on activity, the activity was increased because of the change of bedding. So I'm just mentioning this because when planning, I think Joe mentioned it as well, when planning experiments or studies in which the activity monitoring or the activity levels information is needed to evaluate the disease progression or disease progression, there are certain things that need to be taken into account beforehand to make sure they don't interfere with the evaluation. Now, moving a bit to another aspect, as I mentioned, by using the pancreatic orthotopic model, that gave us the opportunity to assess the possible impact of surgery in the activity levels of our animals. So again, we hypothesized that we could use the activity level measuring as a tool to assess animal welfare with some advantages versus the traditional methods. So by using a 24/7 monitoring, we had access to the night period that is usually missed. We thought that an invasive procedure such as surgery will cause some discomfort in the animal, and that can translate to a reduced home cage activity. We liked being able to have access to the night period because that could give us better insights on how the animals behave. So in our facility, after a surgery day, the animals are obviously checked 4 or 5 hours after surgery, and again before we leave the facilities. But after then, they are alone in the animal unit, so they might be showing signs of pain or signs of discomfort that we are not able to see because we're simply not there. So by monitor activity levels constantly pre and post surgery and comparing those two, that could be used as a tool to identify and evaluate possible refinements in the surgical technique itself or in the analgesia protocols that the animals receive. So those graphs show the activity levels pre and post surgery of males individually housed, and those are 2 different studies run more or less in parallel. The graph, as I mentioned, both show dark and light period activity. The blue line is the dark hours, the red-orange line is the light hours. The gray area between 48 and 60 represents the day of the surgery or the night after surgery if we focus on the dark period. And this type of data allow us to compare not only the quantity of movement or activity that the animal are showing, but also the patterns of behavior that the animals have. And as you can see, after the surgery, we see a decrease in night activity after the surgery. We don't see it as much during the day, but again, during the day, the animals don't move that much to start with, so it's difficult to detect this variation. And what we can also see is an altered pattern of behavior. So prior surgery, the mice seems to be quite consistent of how they move throughout the night. After the surgery, they seems to be a bit more diffuse and not as consistent as prior surgery. We focus then on analyzing the night period, and those two graphs in here show the average movement per night of those animals. The red box is the night immediately after surgery, and we can see a drop in activity after the surgery and a subsequent progressive recovery of activity almost to pre-surgery values. As I mentioned, this is based on individually housed animals. We thought whether we would be able to detect similar changes in group housed animals. In this case, I'm showing the results of females in now, and they were housed of 5 mice per cage. The surgery that they underwent is the same as the males, it's a pancreatic implantation, and the graph represents similar information. Again, prior surgery, we can see that the mice are more active during the dark period, and we can see again this nice pattern of behavior that the animals seem to be reproducing night after night. After the surgery, we see a drop in activity, both in day and night, and we see an alteration of the patterns of activity few days after surgery. When focusing on the average night activity, we see similar that we've seen with the boys, which is a reduction of activity immediately after surgery, and then a slow and progressive recovery of the activity to normal measures. Now, having access to these activity measurements and activity patterns has allowed us to have a better understanding on how invasive procedures can affect the animal welfare, and gives us a tool also to evaluate refinements of the technique, since we have a better understanding of the impact of the surgery in the animals by having access to those moments in the life of the animals that we didn't have access before, which is the night period. Now, at the moment, we are evaluating whether we can reduce the impact of the surgery in those animals. As I mentioned before, we assume that this reduction in activity is because the animals are in some sort of discomfort. So we are evaluating changing the analgesia procedure protocols that we use to the animals. And the good thing is that we can use the same of monitoring the animal movement to see if what we think is a refinement for the technique is actually having an impact on the animal welfare. So to summarize, based on our evaluation, we reckon that monitoring the animal activity 24/7 in the home cage can provide a good tool to monitor the disease progression and the impact of procedures that we do into our animals. So we have shown by using both flank and orthotopic tumor models that the progression of the disease has an inverse correlation with the activity during the night period. This correlation or this understanding can help to increase the monitoring of the disease and the understanding on how the drugs in development can modify the disease progression. And finally, we have shown that assessing the animal activity after invasive procedures, such as the surgery, can be used to have, to have a better understanding of the impact of whatever procedure we are doing to the animals on their welfare, and can be also used as a tool to evaluate possible refinements of the technique. And to finalize, just a massive thank you to everybody that has collaborated in this study. It, as you can see, is a big team with different skills and knowledge, and something that John mentioned before, the amount of data that the DVC provides and the way that that data is treated requires some specialized analysis for that. So thank you everybody for that, and thank you all for listening as well.
Thank you very much, Judit. The clear indication of data use across with the experimental data that you've had as well. So thanks very much. And just as before, it asks a few more questions as a result of this study.
Absolutely.
Very interesting.
Thank you.
No problem. Thank you very much, both of you. So we've got some time for questions. Thanks everybody for attending and listening to those presentations. We've got a question here from Stefano, believe it or not, for you, Judit. Just turn this on. So Stefano said, do you think that increasing temperature after surgery in the holding cage environment would speed up the recovery of the animals? If so, how would you conduct such an experiment?
So yes, that's the answer to the first question. And that is something, when I mentioned that we were evaluating our surgery and analgesia protocol, that was part of the discussion. So at the moment, we are keeping the animals in a warming cabinet after surgery, precisely for that, because the surgery obviously implies anesthesia, and that results in a decrease of the body temperature in an animal that is small and can lose body temperature really quickly. One of the refinements we thought would be possible is to extend the period that those animals are in the warming cabinet or kept in a warm environment. We think that that will help the recovery period. Whether that will have an impact on the night activity, that's what we are evaluating, or not, that's a different way of analyzing that. I guess if we have a way of warming the actual cage in the DVC rack to keep that temperature slightly higher during the night, that will mean that the mice will stay in that warm, probably nicer temperature for the period that we are evaluating the activity levels. But I don't know how to do that.
A conversation for another day, I think. But no, thank you. Thank you. Okay, I've had another question in from Matt. Thank you, Matt, for Joe. Did you look at other time windows in the 24-hour day, or is it simply too much data to process? Hello, Joe.
Hi, sorry, I can't unmute myself, Scott.
Okay, no problem. Did you get the question?
Yes, I did. Yeah, we actually looked at the activity across the whole 24-hour period for these animals over about a week to see, before we started the study, to see which of the time intervals were going to give us the answer to the question that we wanted. And it depends on what statistical package you use. Some are much more precise and they can look at different the whole 24-hour period and give you a much more precise answer, which we actually have done since this presentation was put together, which is why I've said you can look at data and do it in different ways. So we did look at different time periods, but we looked at them to see which time period we felt would give us the answer to this question, because we wanted a really precise answer. And when we looked at, because these mice have such spiky activity periods, if we looked at the whole 24-hour period, you couldn't see any real clear differences, because it's all as an average. That answer your question?
Thank you, Joe. Okay, good question from Stuart. I think this is targeted at both of you, really. So you first, Joe. Activity levels have been used as a metric in both studies. Have you considered more detailed metrics, such as a loss of circadian rhythm robustness? New metrics exist to measure the stability and reproducibility of rhythms, which may be more sensitive to disruption.
Yeah, not for this. I mean, this was one of our earlier studies, and we are actually doing much more testing of the data. The activity levels really the main thing we were looking at for the DVC rather than any other metrics, but it would be great to have a much more holistic overview of what the animals are doing during the day. And we weren't expecting to see this loss of circadian rhythm So that's something we want to explore more.
Okay.
Interesting in any suggestions you've got there.
Thank you. Judith, have you got anything to add to that? Your side?
Yes, I'll put my video on so people can see my face, otherwise it's a bit too cold. So yes, I agree with Joe. I think having access to not only the circadian rhythm, but also the patterns of behavior that the animal will add further information into that. Now, we haven't done this analysis, but we were quite surprised after the analysis of the impact of the surgery. That we saw a nice constant pattern of movement pre-surgery, and post-surgery this pattern of movement was altered. So I think that's in part caused by an alteration of circadian rhythms or just the discomfort that the animals are having. And yes, it would be nice to have a better analysis of this behavior.
Okay, thank you. And then followed up by saying, has the cage position been considered in any of these studies? Light levels are very different at different rack positions, and this will influence activity levels and activity onset. So I suppose to you first, Judith, just to continue.
No, we have not taken that into consideration. However, we have focused our analysis in the night period. At that point, the light is not that relevant.
Okay. Joe?
Scott, yes, we have, not for this study, but for the study that I talk about in another webinar, we did do a statistical test on the cage positions, and that's why we tagged the outliers to see, and there's no correlation between cage position and activity levels in the study we did. Doesn't mean it's not in other studies, because all the animals are different, so it's worth doing that kind of test if you think there may be an influence, but we didn't find one.
Scott, sorry, I wanted to add to the second question about the looking at differences in circadian rhythm robustness, or differences during the day and not just the early level. In ALS study, it's also summarized in the review that it was been written. We look specifically at these things, so really changes of how basically the pattern over time changes, and basically you can see also in Parkinson, Alzheimer, where you expect basically that the regularity changes. We brought a new algorithm that is available to actually monitor those type of changes. It's not just level, it's really how this changes over time, so the chaos of the data in 24/7.
Thank you, Stefano. Another question here from Mark. Have either of you used the DVC to assess single housed animals or only grouped housed? This assessment would an alternative to monitoring in an open field system. I suppose to you first, Joe?
I'm not completely sure what the question is. Do you mean looking at single house versus group, or how they would react in an open field if they're in a single house or a group house situation? We have looked at different, we have done a test on whether there's a linear between different group sizes on the DVC, and we didn't find any linear, so we'd need to, it's important to have, you can't compare a single house with a group housed animal on the DVC, because I'm not sure what the question is, I'm sorry.
That's okay. Mark, feel free to elaborate a little.
Scott, I think I understand exactly what is meaning. Namely, you generally test the activity in an open field. If you want to have a general activity, you test for 10 minutes, 15 minutes, just to have a understandings of what activity between control and whatever treatment you're going to do is different. That's typical in behavior and neuroscience. Also in the review it is described actually what the difference between an open field and repeating actually the open field is versus a DVC. You see you have 24/7, open field is just 5 to 15 minutes according to protocol, and there are differences there. So many people are using actually the DVC to test the baseline activity, and then they use other tests like elevated plasmase and stuff just to test the emotional response rather than open field. Actually, open field has been quite controversial and has been dismissed by many labs recently. So
yeah, I don't think you wouldn't get any difference in the open field on a DVC than you would from an IVC, because it's the same cage.
No. It's, I think, another question. It's really the open field arena versus the DVC. I think, if I do understand correct the question.
Okay.
Yes, and also the electrodes, which Joe pointed out across the cage base, can be singled out so you can compare with an IVC or DVC system the outer edges of the cage with the inner electrodes if you want to compare some similar sort of data. Obviously, it's not an exact replacement, but it may be a refinement anyway. Thank you, Mark, for confirming. Okay, I think that's all the questions. Thank you, Joe, Judit, and Stefano. Very clear, very in-depth presentations with a lot more questions that we can discuss later. Thanks, everybody, for your time on what is a very interesting week. I wish you all the best. Stay well, and hopefully speak soon. Thanks very much.
Thank you so much, Scott. Thanks for organizing this. Really interesting.
No problem.
Yeah.
Thank you,
Day.
Scott.
Thanks everyone for
Thank
listening.
you, everybody. Take care.